This is Dr. Jesse Salk. He's a molecular biologist, clinical oncologist, and a co-founder of TwinStrand. He invented and developed TwinStrand's core duplex sequencing technology with colleagues at the University of Washington. He has authored more than 24 peer-reviewed publications in cancer biology and genomics and has more than 50 issued and pending patents. In addition to his executive and scientific leadership role at TwinStrand, Jesse is an affiliate clinical faculty member at the University of Washington and Fred Hutchinson Cancer Research Center and sees cancer patients part-time at the VA Puget Sound in Seattle. So welcome.
Thank you very much. All right, I think I'm close to the second to last thing between you guys and lunch. Sorry about that. Okay, so Andrew gave a great introduction to cancer overall, and what I'm going to talk about is a bit more the technology and some of the kind of major applications of that technology in that space before early cancer formation. So the next speaker is going to talk about early detection of cancer, and historically speaking, I'm an oncologist, Andrew's an oncologist, we treat patients with late-stage cancer, and that's an important role. But we would all much rather treat cancer at earlier stages. Generally, the earlier one detects a cancer, the more treatable it is. And the talk that follows this is going to be talking primarily about early detection. But I think the best thing that we can do is not treating late-stage cancers, not doing early detection, but doing primary prevention. And primary prevention means doing things that prevent the cancer process from starting or moving along at a given rate. So you've seen this picture a couple of times already today. Cancer takes many years to form. Mutations occur at the beginning that let some cells grow a little bit better than their neighbors if that mutation, that change in the DNA happens at just the wrong spot in the genome, and then things kind of move along and evolve. So things that prevent mutations from happening in the first place, changes to the DNA, or things that prevent or slow down those changes that happen from spreading into other cells, all of that will delay this process. And like Andrew talked about, this process in one form or another is going on in all of us at some very low level. If we all lived to 150, we would unfortunately all get cancer. Cancer didn't used to be that big a health problem, not because it didn't exist, it's because we died of trauma, infection, and malnutrition for most of human history. And so the fact that we've done a good job managing other areas of sort of health and medicine means that cancer is a more relevant problem. And as we're living longer, these processes that are going on become more relevant. Carcinogenesis is a term that means formation of cancer. This entire diagram here is carcinogenesis, the process and the different stages of cancer formation. A carcinogen is a exposure, which could be a chemical or radiation, for example, that somehow contributes. Some of those chemicals directly change DNA. Some of them help cells that have those changes in their DNA kind of avoid the immune system or grow blood vessels. So a carcinogen is a exposure that somehow accelerates this process. And we know that being a firefighter is a cancer risk, and because many things around you guys are carcinogens. Historically speaking, the way we know that being a firefighter is associated with cancer is that there are more cancers in a group of firefighters than other relatively age-matched or other exposure-matched controls. So in a given city, people who are in early 30s who are firefighters compared to someone in their early 30s that lives in the same environment. We know that from experimental evidence, of which we've heard about over the last couple days, you can take some of these chemicals, a polyaromatic hydrocarbon, and expose an animal or some cells in culture. And we say that, well, that kind of makes sense that some of these things firefighters are uniquely exposed to can recreate cancer in model systems. Biological plausibility is another thing we use. We say, well, we know that this chemical directly modifies DNA and that cancers have more mutations in their DNA, therefore this all fits together. And then taking all of this together, we can say, okay, well, this bit of evidence, this bit of evidence, this bit of evidence all comes together, and that's why we have IARC, position right now. Just there's a lot of different evidence. But the challenge is, this is great to know that being a firefighter is a risk for cancer, but that doesn't directly help us act on that. Because within firefighters, there's variable exposures, dose, duration, chronicity, skin versus inhalation, and it's hard to know exactly who gets what and how. Many exposures come in diverse or unknown combinations. In fact, probably the vast majority do. Independent exposures may interact synergistically. So even if you don't have a fire that's burning up 5 different poisonous chemicals at the same time, maybe something that happened 2 days ago actually is synergistic with something that happens later. We know, for example, individuals who are both smokers and exposed to asbestos, that is a synergistic carcinogenic action. If you're a non-smoker and exposed to asbestos, or a smoker and not exposed to asbestos, those 2 risks are actually independently lower than that combination, and that's probably true based on some of the mechanisms of how these chemicals act. And then the last challenge here is that the most realistic experiments for what is actually going on in humans are impractical or impossible with a lot of existing technologies. Yes, we can set a couch on fire inside a trailer, but we're not going to put real humans in there. And so a lot of these things that we do with surrogate experiments, measuring chemicals, measuring levels of chemicals, measuring how well soap extracts chemicals, measuring if those chemicals get into you, that doesn't necessarily reflect exactly what those chemicals are doing at those doses in combinations in individual people, which just comes down to the fact of the matter is, being a firefighter is complicated, and the ultimate reason that cancers are higher in firefighters is a complicated mixture of factors, and they're hard to individually measure all the way through to the level of those earliest stages of cancer. So put this another way, we know that cancer risk goes up with age. We know that individuals that have certain exposures, that risk goes up higher. And so I'm just giving you a theoretical curve here of aging in blue increases your cancer risk, and aging, being exposed to a certain environment, let's call it the firefighter exposure, increases this more. But from an individual patient perspective, you don't know where you fall in that curve, and you don't know if you were somebody in this population that is a very high risk or a very low risk. This is a population average. And so from the perspective of actually knowing about the relative risk for a given individual, or the very specific sub-exposure that is causing the biggest piece of that risk, it's very challenging trying to work through sort of model systems or epidemiology without being able to measure that exact cancer process in a variety of individuals directly. So historically, there have not been very many ways to link an individual's cancer to an exposure. Andrew talked about how it is now possible to sequence the entire DNA, the genome of a cancer, and look for patterns of mutations that correspond to certain chemical exposures. That's not been possible before, but it is now beginning to be possible. Second point is, nobody likes statistics. Saying that your risk is 2-fold higher, 3-fold higher, based on knowing that you're a firefighter is not very helpful. It would be much easier and much more helpful to say your specific risk is 70 times higher, or it's only 0.5% higher, because all of these factors that go into firefighter do not apply to every single person. There's different sub-pieces that may be higher or lower relative risk. If a chemical is known to increase cancer risk, it's hard to know how much exposure one gets. We talked about measuring individual benzoate pyrene levels in urine, but there's a bazillion polyaromatic hydrocarbons. And so measuring one chemical is a surrogate for what's actually going on in that cancer formation process. It's not it's not measuring the process itself. Because somebody has been exposed to a carcinogen, it doesn't necessarily mean they're going to get a cancer. It increases probability, but someone's unique genetics make them somewhat resistant. And if we said you got a really high level exposure, and somebody is taking that as, I'm destined to definitely get cancer, that's not necessarily true, and that can be a really harmful thing to say. So basically, all of this adds complexity to taking ourselves from being a firefighter increases cancer risk to how does that really affect individual people or the individual components of what those people have experienced that let us risk stratify and decide for a given person how high is your risk and what can we do to, knowing you have that risk, to lower your long-term risk, for example, with more increased surveillance. And it also affects our ability to make decisions about how to mitigate things. So kind of high-level summary is one of the largest challenges to exposure-associated cancer risk assessment and development of mitigation strategies is our inability to objectively measure the early impact of a carcinogenic exposure in humans. And there's three operative words. Measure means the ability to actually quantitatively detect. Impact, I'm not talking about the chemical being present, I'm talking about where you are on that cancer formation pathway, where those mutations are and how have they had cells start evolving. And early, meaning it's not helpful to know 20 years later that there are more tumors, you have a cancer 20 years after exposure. We want to be able to measure the early impact, the early part of that cancer evolutionary process so that we can, again, manage individual patients and then manage mitigation strategies for firefighters as a whole. Andrew talked about this already a bit, so I'm not going to harp on this, just going to give a few definitions. You've heard the term genomics. The genome means all the DNA in your cells and some of the elements kind of related to that. Genomics is the study of all the DNA in one's cells. Mutation is changes to that DNA, and the sequence of that DNA is a code, and when that code changes, some of the time it breaks things in cells and lets them become cancerous. A somatic mutation means mutations that happen during your life, not something you inherited from your parents, but something that happened as a result of simply living, and with many types of carcinogens, they induce or increase the rate of somatic mutations. And the last term here is next generation sequencing. This is the sort of contemporary DNA sequencing tools we use. It's a kind of a funny term because you say next generation, well, you can never get to the next generation, right? It means it's the next generation. But basically, when you see this word, this phrase NGS, it basically means what we use for DNA sequencing now. Couple other terms. Carcinogens are things or exposures that increase cancer formations rate. A mutagen is one type of carcinogen. Remember, if you change the DNA in cells, it can make them more likely to turn into cancer cells, but there are also carcinogens that don't act by directly mutating the DNA. They act by kind of helping cells that already have some mutations grow better than their neighbors. So mutagen is a subset of carcinogens. There are also non-mutagenic carcinogens, and this is important for a couple slides later. Standard or conventional NGS lets one look at cancers and look at genetic information and help look backwards. Do what's basically, I use the term archaeology. Archaeology is looking at data to infer what happened in the past. That is useful insofar as somebody who has a cancer, us being able to say, well, that was potentially related to this polyaromatic hydrocarbon. That's helpful, but what would be more helpful is to know that information before that cancer formed. So can we detect these changes, not in a tumor, but at very early stages when those mutations are present in only a few cells as opposed to trillions of cells? So current standard DNA sequencing technologies are good at looking at the tumor and looking backward. They're not good at looking at healthy appearing tissue to look for those early changes related to the question somebody asked earlier. Risk assessment about cancer risk is about the future, not the past. We want to see changes in the genome that are not in a cancer where all the cells have these mutations. We want to look in healthy normal tissue where some of these things are occurring at very, very low levels, and standard DNA sequencing tools cannot do that. And the reason for that is they are not accurate enough. Most technologies or assays that have historically been used for the field of genetic toxicology, which is being able to assess whether a chemical is a mutagen or a carcinogen, something that changes the genome, are really set up using kind of model organisms, some of which Andrew talked about, and they're designed for like pure chemicals, like a new drug. We say we give this to an animal, we see if it can create changes, and to know that this chemical that we're working with in an idealized lab is potentially carcinogenic. Those technologies, which are widely used in drug development, cannot be used in humans because they require these special genetically engineered systems to detect very low frequency changes. There are kind of three technologies that Vinny asked me to talk about that can be used in humans without, but you just got to remember from the perspective of kind of scientific tools people use for looking at mutations, historically the vast majority of even the most sophisticated ones have not been applicable to humans. They've been applicable to model organisms. Mice are not small firefighters. They just aren't. Comet assay is a technique that looks for breaks in the genome, in the DNA, and it's just known that breaks in the DNA increase risk for mutations. It's an indirect way of looking at cancer or mutation risk. It's several steps removed from cancer. PIG-A is an assay that involves a technology called flow cytometry, which sort of looks at patterns of proteins on the surface of one particular cell type to infer the presence of a protein, and if it's not there, it suggests that a mutation happened in this very specific protein. So it's a kind of a cool technology, but it's pretty limited in the breadth of things it can do, but it is a sensitive technique that can measure mutations in humans, but only really in one tissue type. And the last thing is what I'm going to mostly talk about now, which is error-corrected sequencing. This is using new forms of next-generation sequencing. So next generation or next, or whatever. But I'm going to call it error-corrected sequencing. I'm going to specifically talk about duplex sequencing, which is the sort of most accurate form of that and the one that we work with. Standard next-generation sequencing can't detect genomic changes in non-cancer tissue because it's not accurate enough. This is, if one is looking at mutations in a tumor where all the cells contain the same mutation, it works just fine. But looking at 1 in 100,000 cells, it doesn't work because the error rate is, say, about 1%. You can't measure 1 in 100,000 or 1 in a million if your error rate is 1%. So this lower left plot shows you a kind of a simplified version of some genomic data. On the x-axis is nucleotide position. This is a 400 base pair region of the genome. The y-axis is the percentage of molecules being sequenced that have a particular mutation, and each tick along that axis is the a nucleotide position among those 400 sites. And inherited mutations, if you get it, or variants, if you get a copy, something you inherit from both mom and dad, it's called homozygous. It's in 100% of those molecules. And if you inherit it from just one of your parents, it's heterozygous. You see it in half of the molecules. There are small, that little bit of red fuzz at the background is the background of technical errors of the sequencer. And if you zoom in and make the y-axis 1%, you can see that essentially every position in this small region of the genome with normal sequencing appears to have mutations. This level of background noise is irrelevant when you're looking at inherited things. There's no possible way you could confuse them. But if you were trying to see something present at 0.1%, there's no way to tell what's real and what's not. Error-corrected sequencing and duplex sequencing specific is a technology that uses special biochemistry and special kind of informatics to basically get rid of all those background errors and lets you detect very, very low frequency things with high specificity. This is standard sequencing, and this is duplex sequencing. There's only one real mutation present in this patient. The rest was errors. And this mutation, actually, somebody brought up CML earlier. This is a patient that had chronic myeloid leukemia and relapsed several months after the sample was taken, and that was a known drug-resistant mutation that was present in a small number of cells, and had I, as an oncologist, known that, we could have put that patient on a different drug to prevent that relapse from occurring. So just one example of how that level of sensitivity is important. Applying this to, going to firefighter applications or to measuring mutations. Remember, mutations are a necessary part of cancer formation process, and chemicals carcinogens that create mutations, or chemicals that create mutations are called mutagens, and pretty much all things that are mutagenic are carcinogenic. They have some risk of increasing cancer. The way our sequencing process basically works is we sequence about 1 billion nucleotides of the genome with very, very high accuracy. And then we look at how many of those nucleotides have a mutation divided by the total number of nucleotides. So, for example, in this case, if we sequenced 189,664,560 nucleotides and we found 16 of those had a mutation, the mutant frequency would be 8.43 times 10 to the minus 8th, or 8.43 per 100 million. And that is this bar right here, this first bar. So this is data from mice. This is not humans, but this same thing applies to measuring in humans. This is mice that were treated with a chemical called urethane, which many of you are familiar with. It unfortunately is present in all sorts of industrial chemicals, and if you are breathing fumes from a plastic factory that's on fire, one would be exposed to urethane. It's a known mutagen. This is looking in lung, spleen, and blood, DNA that's isolated from those in mice. Each one of these ticks is a different animal. So there was 5 animals treated with either nothing or urethane. And when we take DNA from exposed versus non-exposed lung, spleen, and blood, you can see that there is a significant increase in the mutant frequency. This indicates not just that urethane is present, but it's saying that it's actually changing things in the genome. And this is measuring those changes not 20 years after exposure when a tumor might form. This is measuring it a couple of weeks after exposure, when that chemical has been put in the environment, absorbed, cells take it up, the DNA gets damaged, and then they actually modify the genome based on that damage affecting the ability of a cell to fix its DNA. So that's how a calculation is done. We can take this in any species. This is mice and this is rats. You can see that with different doses, your level of mutations go up. You can see at different time points, we see different tissues accumulate mutations at different rates, somewhat dependent probably on how fast it takes the cells to divide. This is a publication that Stephanie Smith-Rowe in this room here has coming out soon, and this is one that one of Paul White's colleagues worked on. And so we can see in animals, a variety of species, this can be done. We can apply the same exact thing to humans, and we, because the technology just requires DNA, it doesn't matter where it comes from. In addition to measuring the frequency, we can look at the pattern of mutations, or the spectrum. Different chemicals create different types of mutations differently because they break the DNA in different ways. So in the case of urethane, you see a lot more of C to T and T to A mutations for biochemical reasons than you do in untreated animals. And you can simply see, without knowing anything about what this means, the kind of pattern on the left of the relative abundance of different types of mutations is very different than the one on the right. A computer, just saying which is most similar to what, was able to separate these with perfect sensitivity and specificity without knowing which animals were treated or not treated. And so these patterns let you not only identify exposure with even higher fidelity than looking at that kind of just overall mutations induced, it also gives you some idea of the mechanism. And if you see a particular pattern of mutations, you have a better ability to say, not only is this person's genome changing, and not only is it changing relative to an unexposed population, I can say kind of what the chemicals involved might have actually been. One can get even more specific and not look at just the subset of mutations that are A to T or C to G. We can subdivide it even more, and you can subdivide each one of these mutation types down based on the 2 bases, the 2 nucleotides that are adjacent to that sequence. Those bases nearby modulate mutagenic potential, and this creates what's called a trinucleotide signature. And these signatures or patterns are very, very distinct for different chemicals. Benzoate-pyrene, one of the polyaromatic hydrocarbons we talked about earlier, urethane, which I talked about here, and ENU, which is an alkylating agent. These all give very different patterns, and a computer can look at these patterns and say, this corresponds to this signature, that signature, and it's likely related to a particular chemical. We've been able to sequence tumors using standard DNA sequencing and look backwards, archaeology, and say, this tumor probably was partly caused by this particular mutagen that has this pattern, but what this technology, duplex sequencing, can do is look in normal, healthy tissues within days or weeks after exposure to see that. So this pattern that we see at very low levels is the same pattern that we see, say, for benzoate-pyrene. It's the same pattern you see in lung cancers from smokers who were exposed for 20 years. We can see the same thing after a few days. We can apply this in humans. There's a chemical called aristolochic acid, which is a mutagenic chemical that's in a particular type of traditional Chinese medicine that causes liver and kidney cancer. And we can look in blood and urine and identify not the cancer itself, but the fact that an individual is exposed. So in this case, I could say, we know this, people who've been exposed to this chemical have a much higher risk of cancer. For an individual patient management perspective, I could say we should be doing regular screening for these patients at much higher level than an average population because they're at much higher risk. So that helps individual patients who do not yet have cancer be able to catch it earlier. But it also, from a broader public health perspective, we can say, everybody in this town or this little village seems to be exposed, and people in this village in Taiwan don't seem to be exposed. Let's do source control. We look in Firehouse A or Firehouse B, and we say, you're doing really good decon here or not, or there's nastier chemicals in the fires near you. So for both individual patient management as well as broader interventions, having that information is really relevant. Last thing I'm going to talk about is that not all chemicals that are carcinogenic are mutagenic. Some of them operate by not directly changing the DNA. They help the cells that already have some of these changes that happen as a normal part of aging grow out. And so one other thing that we can do is you can use this high accuracy DNA sequencing method to actually see the effect of non-mutagenic carcinogens, which is very counterintuitive. How are you doing DNA sequencing to look for something that's not directly changing the DNA? Well, what we do is we look at genes or parts of the genome that are known to be able to create cancer, that you see them in tumors that form, and we can look for the presence and expansion of those. We see them instead of at 1 in a million, or 1 in a million, we see them at 1 in 1,000. And that basically is letting us see what are the microscopic precancerous clones growing out. So we can assess not only mutagenic chemicals, we can measure non-mutagenic ones. And many of the things that firefighters are exposed to fall into both categories, and some actually operate through two means. Not going to go through this in detail other than to say this was a study in a particular type of mice that it's known if you paint what's called an initiator followed by a promoter on their skin, they will get cancer. If you use just the initiator, they don't get cancer. If you use just the promoter, they don't get cancer. If you do the promoter followed by the initiator, they don't get cancer. But if you do the initiator followed by the promoter, they do get cancer. And it's known that now, in modern times, the initiator is a weak mutagen, and the promoter helps those mutations expand in a way that they don't in normal tissue. So if we look at vehicle, which means untreated, this first chemical, DMBA, which is the initiator alone, or the DMBA followed by TPA, if you look at mutation patterns that are induced, you see nothing unusual in the untreated one. You see mutations in the one with the weak mutagen, as well as the one with the weak mutagen, followed by exposure to this promoter. And that's kind of what you would expect. But if you look in some genes that are very specifically required for cancer formation, what we see is in the untreated ones, we don't see any clones of cancer driver genes, so no kind of microscopic genetically definable foci of tumor. If you look in the initiator without the promoter, you don't really see any of those. But if you see the initiator followed by the promoter, you see all these microscopic, molecularly defined clones of cancer driver mutations that start appearing. And so we are measuring two different things entirely that are highly, highly cancer relevant. And you can see this in a way that's just never been possible before. This can be done directly in humans. So in summary, historically, relating exposure to cancer risk has been epidemiologic at a population level. It's helpful to know being firefighter is a cancer risk, but it would be much better if we could be more granular and say how much each person's individual risk is and what specifically is creating that overall risk. Standard DNA sequencing lets us look backwards. So in firefighters who do develop cancers, we can take those cancers and try to identify what caused them, but that person still has cancer. It's helpful for colleagues. It's not as helpful for that person. Newer sequencing technologies, we can identify these changes that are not just chemicals, the presence, but actually the thing that the chemical is doing, that in that earliest stage of cancer formation, we're measuring that early carcinogenic process in a way that lets us assess dose, exposed tissues, potential the type of compound, even where the compound in the tissue, where in the individual the mutations or cancer changes are happening. It's not necessarily uniform. And we can do this for both mutagenic and non-mutagenic carcinogens. I'd shown this slide last year, and this is just a list of some ideas, and this could be something we could talk about later at the working sessions of what potential ways these tools could be used to look at various aspects of firefighter cancer risk in a way that is potentially actionable. As an oncologist and anybody in medicine, one of the things you always think about is if you measure something, what are you going to do with that information? And it's really important to say, if I had this information, I would change a process. I would change screening surveillance intervals. I would make people do a certain type of decontamination. I would advocate for certain chemicals being transported a different way. And these are some ways that one could potentially do experiments to inform those decisions. So there's a summary. These are June and Aaron's contacts on here, who you guys have met before. Contact them. Happy to answer questions and talk to you during the break. We'd love to work with you. Yeah.
So I have a real quick question. Any analytical technique is as good as its sampling method, right? So if you have one cell that has this mutation among billions that doesn't have mutation, how do you confirm, how do you make sure that you get that cell that has the mutation to analyze?
So yeah, you heard the question, which was, your sampling method is really important. Even if you have the most sensitive tests in the world, how do you know that you're going to sample the cell that has that mutation? The reality is these things like mutagenic chemicals, they create all sorts of mutations. Each cell has 3 or 6 billion of those A's and G's and T's and C's in it. We sample millions of the cells, and the idea is that we're trying to sample enough of the genome to have a good chance. So the reason in the example I showed you, you sequence of 200 million nucleotides is, you're right, if I only sampled enough to see 1 versus 0, you're not going to have a great chance of measuring it. We try to measure hundreds of times more than we need, so we're saying 20 out of a billion versus 200 out of a billion. So you require sampling a lot, which means sequencing a lot, which is now possible with these new sequencing tools. It's a great question. Yeah, so the question was, I brought up promoters, and the question was, is it possible that reactive oxygen species can act as a promoter? And the answer is yes. And the more complicated, nuanced answer to the simplification that I said earlier is there's non-mutagenic carcinogens and mutagenic carcinogens. There's actually a continuum between them. Reactive oxygen is a great example. It can directly bind to DNA and mutate it, but it also kills healthy cells. And when healthy cells die, there's space for slightly abnormal cells to grow in. So the answer is mutagenic chemicals or anything with toxicity including reactive oxygen, can act as a promoter as well. We know chronic inflammation and a lot of human diseases, autoimmune diseases, other things are risk factors for cancer, and a lot of those show reactive oxygen. The question was, what is a skin painting model? A model system is something that is a experimental surrogate that sort of represents something, and so that's what a model system is. And in this case, it's a particular type of mouse, and the experiment is one takes a chemical and uses a paintbrush to literally put some of this chemical on the skin and let it sit there, and then follow that by putting on a different chemical. So we wouldn't do this in humans because these are carcinogens, but it's a way to, in animals, study what creates those cancers and what we could do to potentially slow that down. And I showed you how we can measure that with this DNA sequencing method, but we could do the same thing. We wouldn't paint a carcinogen on a firefighter's skin. We would look at the DNA of a firefighter's skin who is exposed versus not exposed. All right. Do one last one.
Sure. I'm sure that the differences in course of treatment would be very different for precancerous diagnosis. So could you just kind of tease apart what that might look like?
So the question was, if you have this information, what do you do with it exactly? Because how you treat a late-stage cancer is very different, presumably, than an early pre-cancer. And that's a really good point. Late-stage cancers we treat with very poisonous chemicals, often which are mutagenic themselves, because the benefit outweighs the harm. We would never treat somebody who has an early pre-cancer with those chemicals because the harm would exceed the good. So this is a balance in oncology in general, toxicity versus risk. Being able to measure these changes doesn't mean we're going to give somebody chemotherapy, but it might mean we're going to do a skin exam 3 times a year or 4 times a year in you compared to a skin exam in an average risk person, which we'll do once a year. Or we'd say we're going to do colonoscopy starting at age 30 in you as opposed to age 50. We could say we're going to ask GRAIL to do a non-invasive blood test to look for early cancers starting at a given age or happening more frequently in a person that's at higher risk or lower risk. So there's a lot of things we can do that are not directly harmful with that information.